Okay, so now we come to the exciting bit of this evening. Well, that was quite fun as well. But tonight's main attraction is our lecture by Professor Rebecca Kilner from the Department of Zoology in Cambridge. And Rebecca is an animal behaviorist and an evolutionary biologist. And I was trying to remember where I met her. And we realized now, she reminded me, that we spoke at the Royal Institution together many, many years ago. And she's looking at how behavior influences evolution. And what she'll talk to us tonight is about burying beetles. But I just wanted to share with you something that I found from her website, which is, I think, has got to be one of the most interesting paper titles I've come across in about the last 10 years. And this was published last year, and the title of the paper is Strategies for Managing Rival Bacterial Communities, Lessons from Burying Beetles. Who knew? But tonight, she's going to talk to us about life after death, the ecology and evolution of burying beetles. Welcome, Rebecca.
So I'd like to start by thanking the Society for inviting me to come and speak here today. It's an honour to address such an illustrious audience and in such an illustrious location. So although this is a talk about burying beetles, the grand theme of the talk today is about the relationship between evolution on the one hand and behaviour on the other. And I think we could say that one of the great triumphs of the last 50 years or so work in animal behaviour has been to show that behaviour can evolve just like any other trait. So there's now a substantial body of theory and experimental work showing beyond any doubt at all that animal behaviour in nature is optimised by natural selection, it evolves, and it is adaptive. So, evolution changes behaviour. But what I want to think about today is what happens next. Because evolution doesn't just stop once an adaptation like behaviour has evolved. And in fact, that adaptation might very well contribute to the way in which evolution then unfolds. So this is the question that I want to try and address today, and that is, how does behaviour influence evolution? Now, of course, this isn't a new question, and Darwin himself led the way in thinking about how behaviour might drive evolutionary change. And he described two ways, really, in which behaviour might do this. And the first is that behaviour can exert selection on others. And he spelled this out in this very famous passage from The Descent of Man, written in 1871, where he says, courage, pugnacity, perseverance, strength and size of body, weapons of all kinds, musical organs, both vocal and instrumental, bright colours, stripes and marks, and ornamental appendages, have all been indirectly gained by the one sex or the other, through the influence of love and jealousy, through the appreciation of the beautiful, and through the exertion of a choice. So what Darwin is saying here is that all of these morphological traits are the result of selection imposed by the behaviour of another member of the same species. And then the second way in which Darwin imagined that behaviour might change evolution was spelt out in his last book, which I'm sure you've all read from cover to cover, The Formation of Vegetable Mould Through the Action of Worms. And here he presents evidence that earthworm behaviour, and specifically their tilling of the soil by passing it through their guts, changes micronutrients in the soil, changes the microbiology of the soil, and so facilitates plant germination, and can even cause dramatic physical change in the landscape. And so as a result of all these observations, Darwin concludes it may be doubted whether there are many other animals which have played so important a part in the history of the world as these creatures. So here we have a modern view of the evolutionary process, where we have variation in ecological conditions, imposing selection on inherited variation, and bringing about trait change through genetic change. And what Darwin is saying here is that behaviour can influence these two key steps in the evolutionary process. It can influence ecological conditions and in turn exert selection, but it can also exert selection directly on other animals. So you might be thinking, well, if this is all known in Darwin's day, why should we bother to think about it again now? And the reason is that we have much better developed theoretical reasons for supposing that behaviour can profoundly influence the evolutionary process. So this is work that is focused particularly on social behaviour, so interactions within and between species. So we have ideas of Eco-Evo feedback, where we have behaviour changing ecological conditions, and then those changed conditions then exerting selection back on the animal that changed it in the first place. We have the idea of social selection, where behaviour means that animals can act as agents of social selection on conspecifics. And we have the idea of parental effects, providing an alternative mechanism to genes for trait inheritance. So what I want to tell you about today is some experiments that we've been doing to investigate exactly how social behaviour changes evolution in each of these three key steps. Now, we can't possibly analyse the huge diversity of social behaviour in our lab, so we choose to focus particularly on parental behaviour. So if we want to study how parental behaviour affects evolution, then ideally we want to study species that's locally abundant, that shows parental care, that breeds readily in the lab, but which has a sufficiently short generation time that we can carry out some experimental evolution. Unfortunately, precisely such an animal exists in the shape of the burying beetle. Now, the natural history of the burying beetle was first made famous by the great French naturalist, Jean-Henri Fabre, and here's the great man himself looking drained by the sheer magnificence of his prose, because here is what he had to say about the burying beetle. The most vigorous and famous of these expurgators of the soil is the burying beetle, so different from the cadaveric mob in dress and habits, for he is a gravedigger, a sexton. He buries a corpse entire on the spot in a cellar, where the thing, duly ripened, will form the diet of his larvae. He buries it in order to establish his progeny therein, and this is the best bit. He makes a clearance of death on behalf of life. And we might as well all go home now, because it's not going to get any better than that. But here, in much more prosaic terms, is the life cycle and the natural history of the burying beetle that Fabre was alluding to. So the process begins with the burying beetle flying around, searching out the newly dead. If they get lucky, they find a fresh carcass like a small mouse or small songbird, and then a partner will arrive, and together they set about converting this dead body into a nest for their offspring. So they shave off the fur or feathers, they roll the flesh up into a ball, and they bury it below ground. And all the while they're doing this, they're smearing the flesh with exudates from their mouth and from their anus. And then while they've been busy doing that, the female has laid her eggs in the soil surrounding this buried carcass. And after a couple of days, the eggs hatch, and the larvae crawl to the carcass, where they take up residence on this edible carrion nest. And they feed on the flesh themselves, but their parents also feed them too. And then within about a week of hatching, there's absolutely nothing left of the dead body except for a few bones. So at that point, the parents fly off in search of new mating opportunities, and the larvae crawl away into the soil to pupate. And that takes a couple of weeks, and then an adult emerges, and then a couple of weeks after that, they reach sexual maturity, and the whole process starts again. So you can see this is a wonderful animal. It's locally abundant in the woods surrounding Cambridge, so it ticks that box. It displays relative elaborate parental care, it ticks that box, and it has a short generation time that makes them absolutely ideal for evolutionary analyses of parental care. So this is the setup in our lab so that you can imagine the details of the experiments as I go through them. This is one of our study sites at Wycombe Fen, just north of Cambridge. So we catch beetles from nature and we bring them into the lab, and there they get assigned their own personal little plastic box with a unique identity label on, filled with soil. And then when we want to breed them, we put them in a larger plastic box lined with soil, furnished with a dead mouse, pop in a pair, put the lid on, put it in a cupboard to simulate underground conditions for a week, and then we come back about a week later and we can measure the number of larvae that they've produced, and we can weigh the individual larvae to get an idea of fitness. And so with this setup, we can manipulate ecological conditions in and around the carcass, we can impose selection if we want to, and we can do the whole thing on a grand scale with many, many individuals, which means that we can carry out evolutionary experiments in the lab. All right, so I'm going to start off by thinking about how behaviour can influence ecological conditions. And for this part of the talk, we're going to be focusing on events before hatching, during carcass preparation. And the work I'm going to describe was carried out by my postdoc, Anna Duarte, and these illustrious gentlemen from the departments of genetics and biochemistry in Cambridge. So before we get going on the beetle behaviour, I thought I should show you what happens to a dead mouse in the hours after its death if it's not lucky enough to get found by a burying beetle. And it turns out there's a surprisingly large number of videos like this on YouTube, so this is where I've taken this from. So we're looking at time-lapse footage of what happens straight after the mouse has died. Oh my goodness, I wasn't intending to have sound effects. So what you can see here is the abdomen of this dead creature, and it's starting to distend. And it grows larger and larger and larger. And the reason it's distending is because the microbiota in the animal's gut are proliferating like mad. And eventually the pressure within the abdomen grows so large that the abdomen ruptures. There we go. And the fluid seeps out. And this is a critical moment in the decomposition of the mouse because this triggers the spread all over the body of the mouse of microbes like the fungi living in the soil and other microbes already living on the carcass. And you can see that illustrated here by the spread of this fungal mycelium. So think about this from the burying beetle's point of view. If it wants to use those resources on the carcass for its own reproduction, then it's got to somehow prevent rupture of the carcass, because that's a key moment in the conquest of the carcass by these rival microbes. And it's got to have some way of dealing directly with this threat of the microbes themselves. So what we're going to do now is look at footage taken from our lab, looking at how the beetles solve each of these two problems. So what we're looking at here is a pair of beetles, and they're going to prepare a carcass body of a mouse for reproduction. And it's about 2 days' work condensed into 30 seconds or so, and it's filmed under infrared light, so there's minimal gore. So what you can see is that first off, the beetles home in on the abdomen, they make an incision, they pull out the guts, and they consume them. So immediately they've dealt with the issue of rupture because they've taken out the guts. And then you can see that they shave off the fur. You can see this white stuff accumulating on the surface of the soil. And then together, they set about rolling it into the ball that will become the carrion nest for their young. And if we'd given them enough soil, this would be below ground by now. And so you can see by the end of this process, what we end up with is this beautiful sphere of flesh glinting slightly in the light because it's covered so completely in exudates from the mouth and anus of these beetles. And it's the exudates that solve the second problem and deal directly with the microbial threat on the carcass, because it turns out that these anal exudates have antibacterial properties. So here's a close-up of the beetle's abdomen, so you can see the anal exudates in all their glory. Now, it turns out that beetle researchers have been knew for a very long time that beetles probably had this antibacterial capacity in their exudates. But when they searched for what might be causing this in the exudates themselves, they could find very few active ingredients. So that was a bit of a puzzle. And my postdoc, Sheena Cotter, solved this puzzle by realizing that the antibacterial activity of the anal exudates is in fact only upregulated when the beetles reproduce. And you can see that from this graph here. So here we have a time course of reproduction. This is where we pair them up at day 0. And this is a measure of the antibacterial activity in the anal exudates, measured by their capacity to lyse bacterial cells. And you can see that when we pair them up, it's close to 0. But as we go through the course of the reproductive period, it rises, it peaks around about the time that the parents are provisioning their larvae, and it falls off thereafter. And Sheena was able to go on in subsequent experiments to show that the reason why the beetles only upregulate the antibacterial activity of their anal exudates during reproduction is because this imposes a fitness cost on the beetle. So they can't afford to do it all the time. They only do it when they need it during breeding. Now, in more recent work, we've been interested in identifying some of the compounds in the anal exudates that confer this antibacterial activity. So we exploited the fact that the antibacterial properties are only expressed during reproduction to identify which genes confer this antibacterial phenotype. So we did that by comparing transcriptomes, so profiles of gene expression from the gut of a breeding female and a gut of a non-breeding female. And because of the lytic properties that we'd focused on initially, we started off by concentrating on the lysozyme genes. So the results are shown here. It turns out bearing beetles have got 6 lysozyme genes, as shown here. And you can see that for 5 of them, there's absolutely no difference at all in their expression, whether the beetle is breeding or not. But for this gene, lysozyme 6, there's a whopping increase in its expression when the beetles reproduce. So that strongly implicates this gene product as one of the active antibacterial components in the anal exudates during reproduction. And Anna was able to provide further evidence to support this idea by carrying out qPCR. So what she did was she looked at individual females. Each of these dots corresponds to a separate individual female. And she looked at the relative expression of that female's lysozyme 6 gene, and then she measured the corresponding lytic activity of the anal exudates. And so what you can see is that there's a nice positive correlation between the two, and this is really rare evidence linking the expression of a single gene to its phenotypic function. But what you can also see is that there's a lot of variation that's unaccounted for, so clearly this isn't the only thing that's doing this job in the anal exudates, and so what we're doing now is trying to find out what else is there that's conferring these antibacterial, antimicrobial properties. So what we wanted to know next was, what do these antibacterials do to the bacterial community on the carcass? So to answer that question, Anna carried out the following experiment. It has 3 treatments, and she replicated it 6 times using soil from 6 different field locations where we trap beetles. So in the first treatment, she filled a box with soil, she put a dead mouse on it, and then she came back 24 hours later and she sampled the bacterial community on the carcass. In the second treatment, Anna started off in the same way, but then at 24 hours, she added a pair of burying beetles. So they went ahead, they buried the carcass, and they completed carcass preparation, but their larvae hadn't hatched by this time, and this is when she sampled the bacterial community on the carcass. And then in the third treatment, Anna decided to play the part of the burying beetle herself. Now, she didn't go so far as to apply any anal exudates or to shave the mouse, but she did bury it, and then she came back at 72 hours and she sampled the bacterial community on the carcass. So the first thing she did was to measure the bacterial load in each of these 3 different treatments. And what we expected to see was that the bacterial load would be much smaller in this treatment, where the beetles are adding their antimicrobials to the carcass, and it would be much greater in each of these 2 treatments. But to our complete and utter astonishment, that's not what we found at all. In fact, we found the opposite. You can see here are the 3 treatments. The mouse sat on soil for 24 hours, the beetle-prepared mouse, and the mouse buried by Anna. And here's our measure of the bacterial load. And you can see it's the beetle-prepared mouse that actually has the greatest bacterial load, much greater than in either of the other 2 treatments. So that was a bit of a surprise. And so our next step was to try and understand how this had come about. And we decided to do that by looking in more detail at the bacterial community on the carcass. So we did that by sequencing the 16S ribosomal RNA genes. So I'm sure you know these genes are unique to bacteria. So that meant that we could separate out bacterial DNA from mouse DNA and beetle DNA. And they're also hypervariable, so they offer a way of identifying different kinds of bacteria by a kind of DNA barcoding. And so the results of that analysis are shown here. It looks a bit involved, so I'm just going to walk you through it very slowly. So this is one sample profile from one replicate. Here are the different treatments. First of all, this is the community on a mouse that sat on the soil for 24 hours, community on a beetle-prepared mouse, and the community on a mouse buried by Anna, and here is the community in the soil for that replicate. And then each of these colours corresponds to a different class of bacteria. So what I want you to notice, first of all, is that when we bury the mouse in the soil, it immediately acquires a large amount of these gamma proteobacteria. there in the beetle-prepared mouse. And so we think that's because the beetles are weeding those out using those antimicrobial exudates that I just told you about. But what you can also see on this beetle-prepared mouse is that there are three classes of bacteria that aren't represented elsewhere. And they're the actinobacteria, clostridia, and flavobacteria. And these are bacteria, the actinobacteria in particular, are found in the soil, but the clostridia and flavobacteria, more interestingly, come from the gut of the dead mouse. So you remember when the beetle prepares the carcass for reproduction, it eats the guts and the microbiome within, and then it passes all the way through the beetle's gut, and some of these come out the other end through the anal exudates back on to the surface of the carcass. So the beetle is replanting these bacteria from elsewhere in the mouse. Now, other labs have shown that the beetle also carries its own bacteria and it seeds the carcass with those as well. And so we think it's this replanting and seeding activities that explain why it is that a beetle-prepared carcass bears more bacteria than either of the carcasses in our other two experimental treatments. So what we want to know now, of course, is whether these changes in the carcass bacterial community are adaptive from the beetle's point of view. And to try and answer this question, my postdoc, Darren Rebar, focused particularly on the bacteria that were derived originally from the dead mouse, from the dead mouse's gut microbiome, in fact, and replanted during carcass preparation on the surface of the carcass by the beetles. And so to carry out this experiment, he used a tool from medical science, which is the germ-free mouse. So these are remarkable mice that are bred at vast expense to carry no microbes whatsoever anywhere in their body. And we were lucky enough to get hold of some of these dead germ-free mice, and we bred our beetles on them, and we compared the larval performance of beetles bred on these germ-free mice with control mice. And because we were interested particularly in the gut microbes, we cut the carcasses in half, so we had a head portion and a tail portion, so we could isolate any particular benefits derived from the gut microbes. And then we bred all of these beetles in regular soil, and then to assess their performance, we looked at this known fitness correlate in burying beetles' larval mass dispersal. So the results of that experiment are shown here. And the first thing I want you to focus on is this graph here on the left, and that shows the data that we collected for the entire experiment. So there are all four treatments here: the control head, shown with the filled blue circles, the control tail, shown with the open blue circles. So these are from the mice that bore germs. And then the germ-free treatments are shown in red, the head portion filled in, the tail portion shown with an open circle. And the thing to notice here is that there's a pronounced negative relationship between the number of larvae on the carcass and the mass of the larvae attained at dispersal. And that's not very remarkable in itself. It simply means that when you've got a lot of mice, a lot of larvae on the carcass, there's less for each individual beetle. So that means that they attain a smaller mass by the time they disperse away. But what that means in practical terms is that if we want to understand how the microbes are affecting larval mass, we have to control for larval density. And so that's what we've done over here, and then some interesting patterns start to emerge. So here on the left, we have the control germ-laden beetles, mice, and over here we have the germ-free breeding beetles. And so what you can see straight away is that when the beetles breed on a germ-free mouse of any sort, they attain a smaller mass, the larvae attain a smaller mass by the time they disperse away, and this effect is particularly pronounced for those that bred on the tail portion, where the guts should have been filled with microbes. So I think what this experiment suggests is that the beetles are somehow adapted to the presence of microbes from the mouse, and particularly from the mouse's gut microbes. And we don't know yet what that adaptation is. We don't know whether the microbes are directly helping the beetle or whether the microbes simply prompt the beetle to provide more care to its offspring. But whatever the mechanism, the beetles are adapted to the presence of the microbes on the carcass, so when we take them away, the beetle performs less well. So what this work shows in general then is that the beetles are changing the ecological conditions on the carcass by restructuring the bacterial community upon it. And what's more, the structure of the bacterial community has important consequences for burying beetle fitness, and so it imposes selection back on the beetle itself. But what I want to do now is to move on and think about more direct ways in which behaviour can be an agent of selection. And the work I'm going to describe now is carried out by a recently graduated PhD student, Ben Jarrett, and a postdoc, Matt Schrader. And now we're going to turn to events after hatching. So I thought I'd set the scene with some home movie of burying beetle family life. So what we're looking at here is the former mouse in a previous life, now become the carrion nest. And you can see snuggled in there are the larvae. And here you can see them munching away, self-feeding on the carcass. But from time to time, what we see is that a parent will arrive and seek out individual larvae, see her mouth parts making direct contact with the mouth parts of this individual larvae, and she's transferring fluid directly to that individual larva. And because of this kind of remarkable behaviour, burying beetles are often described as an insect that provides parental care. But in fact, it turns out that the amount of parental care that the parents supply is actually incredibly variable from family to family. So some families, the larvae are lucky, they have a diligent parent like this who'll look after them throughout the period of development. But other families, the parents might fly off before their larvae have even hatched. And then the larvae have to fend for themselves on the carcass. And they can do this. They can rely totally, they'll be totally self-sufficient and feed themselves on the carcass. And so we exploited this quirk of burying beetle natural history to ask, does selection by parents drive evolutionary change? So what we did was we went and got a whole load of wild-caught individuals and we brought them back to the lab and we set some aside for later measurement. But most of them we subjected to some experimental evolution. So we set up four experimental populations. And for two of these populations, we cast them into what we call the full care treatment. So here we allowed the parents to stay with their offspring throughout development and nourish them and feed them, as I've just shown you in that video. But then we also had two populations that we called the no-care treatment. And here we allowed the parents to prepare the carcass, but as soon as the carcass was prepared and before the larvae had hatched, we whipped the parents out so the larvae had to fend for themselves on the carcass. And then initially we repeated this treatment for 6 generations, and then we couldn't wait to see what happened, whether there'd been any effect of these different parental regimes, whether it caused any evolutionary change. So at this point, we put the whole lot through a common garden environment so everyone was exposed to full care, so we could eliminate any residual environmental effects and be certain that any changes we'd found were true evolutionary change. And then we tested to see whether the individuals from these different kinds of lines had become adapted to that kind of environment. And so we did that by exposing individuals from the full care lines to both a full care environment and a no care environment, and individuals from the no care lines to both a full care environment and a no care environment. And so our reasoning was that if there had been adaptation, we should see that the individuals from the full care line should perform better in a full care environment than in a no care environment, whereas the individuals from the no care line should now perform better with no care than with full care. And pleasingly, that is exactly what we found. And you can see that from these data shown here. So here's the care to the current brood, and these are the different lines that we tested. So the individuals from the full care lines are shown in blue, those from the no care lines are shown in green, and here's the breeding success. And you can see, sure enough, the individuals from the no care lines perform better in the no care environment than individuals from the full care lines, and individuals from the full care lines perform better in the full care environment than those from the no care lines. There's clear evidence for social selection by parents because individuals in the two types of population have somehow adapted to these different regimes of care. So what we wanted to do next was to work out exactly what traits had adapted. And obviously these could be traits in the parents, or they could be traits in the offspring, or they could be traits in both of them. So we allowed experimental evolution to continue. It's still going to this day. We're now in generation 35. And along the way, we've been carrying out experiments to identify new adaptations as they have evolved in response to these different well illustrated by the biting of an incision in the carcass. This is the final act of carcass preparation. Parents bite a hole in the carcass to facilitate entry into the carcass by their larvae. And the individuals from the no-care populations have accelerated when they put this in. They do it much quicker. And you can see that from these data shown here. So you can see prior to hatching in the wild-type populations and in the full-care populations, roughly a third of the population get round to putting this hole in. Whereas in the no-care populations, twice as many individuals now get round to putting this hole in. And we've shown with experiments that this promotes larval survival in the absence of care, so it's not surprising that they have evolved to do this. We've also found evolutionary change in the larvae. And one key way in which the larvae have changed is in the relative size of their mandibles. Now, the mandibles are key to larval survival without parents. They're the way that the larvae bite their way into the carcass and consume the resources without their parents being there. And so perhaps not surprisingly, these are now relatively larger in individuals from the no-care populations than they are in individuals from the full-care populations. And we think that this enables the larvae to be better self-sufficient in the absence of care. And then most recently, what we've discovered is, or what we've been thinking about, is whether the larvae might have evolved to start helping each other if we take the parents away and they're unable to help them. And so to test them, we ran this experiment that's illustrated here. So we started off by giving mouse carcasses to stock beetles. So these have nothing to do with experimental evolution. We just keep them in the lab for experiments like this. And they prepared a carcass, and we took it away from them before they had a chance to bite an entry hole. So we had a standard carcass environment. We added the hole ourselves. And then we created a sibling environment. And there were two different kinds of sibling environments. Either the sibling environment could be comprised of siblings from the full care line or siblings from the no care line. And then we wanted to test how our focal individuals got on in these different sibling environments. So we had a mixture of individuals from different families, from the focal lines and also from the no care lines. So what we end up here with is 4 different treatments, all with 10 larvae on the carcass, where we can look at how full care individuals get on in a full care environment or in a no care environment, and we can look at how no care individuals get on in a full care environment or a no care sibling environment. And we just left the larvae to fend for themselves. There were no parents in this treatment, and we measured how well the siblings got on with each other by looking at how heavy they were at the point of larval dispersal. And then the way that we plotted the data is shown here. We put the mean mass of the bunch of focal larvae from a brood on this axis, and the mean mass of the sibling larvae on this axis. So that means that we can look at the effect of the sibling larvae by assessing the focal larval mass, and we can look at the effect of the focals by looking at the mean mass of the sibling larvae. And if they have a high mass, then we conclude that the larvae are being more cooperative because they're helping each other gain weight during development. But if they have a lower mass, then we would conclude that they're being more competitive. And this is what we found. And what you can see is that the data cluster into the 4 different experimental treatments. So here we have the full care focal larvae in a full care sibling environment, the full care focal larvae in a no care sibling environment, the no care focals in a no care sibling environment, and the no care focals in a full care sibling environment. So the first thing you can see is the larvae that get on best of all are the no care focals in the no care sibling environment. They are all very large indeed. But the no care focals do much worse if they have full care siblings and not no care siblings, because their mass is much lower. Conversely, the full care larvae in the full care sibling environment do least well of all. These have the lowest mass on average, but we can make the full care focals do better by giving them no care siblings. So what this experiment shows is that the no care individuals have evolved to be nicer to each other, to help out their siblings, to help them gain mass, whereas the full care focals have evolved to be more competitive because any larva that's in their environment will gain less weight. So just to sum up what we've seen in that section then, parents definitely are agents of social selection. When we remove post-hatching parental care, We see evolved change in the parents themselves because they front-load parental care to a greater extent, but this also drives change in their offspring, in the offspring's morphology and in the extent of larval cooperation, because the larvae evolve to cooperate more with each other when they don't get care from their parents. And so in the final section of the talk, what I want to turn to now is how behaviour can influence inheritance of variation. And this is work that was carried out by Sonia Pasquale and Ben Jarrett. And the trait of interest that we're going to focus on, the trait whose inheritance we most want to understand in this case, is adult body size. And as you can see from this picture, there's huge variation in adult size within natural populations. And it turns out that very little of the variation that we see in adult size is due to genetic inheritance. And in fact, in this species, the heritability of body size is remarkably low. It's not significantly different from zero. And instead, most of the variation in adult size depends on the developmental environment. So larvae that eat a lot become larger adults, and this is the main source of variation in adult size. So now you're probably thinking, well, if body size is strongly determined by the environment, then how on earth could it ever be inherited? And the answer to that question lies in the phenomenon of telegony. So I don't know how many of you are aware of what telegony is, but this is the definition. It's when offspring inherit characteristics of their mother's previous mates, even though those mates are not the true sire of that offspring. Now, before we knew anything about genetics, before Mendel, way back in the 19th century, telegony was a widely accepted mechanism for inheritance. And it was so respectable that it was in fact mentioned by Darwin in The Origin of Species, who cited the famous story of Lord Morton's mare. So the story goes that in 1820, the 16th Earl of Morton sold a chestnut-brown mare to his great friend Sir Gore Ouseley. And Sir Gore then mated this mare with one of his own black stallions. But to their utter astonishment, the foal that they produced looked like this. And the two men were particularly amazed by the fact that this foal bore black stripes on the neck and on the legs. And so you can imagine their astonishment. Oh my goodness gracious me, they must have thought. How on earth could this have happened? And then Lord Morton remembered that he had previously tried to mate the mare with a quagga, which looks like this. And so naturally he concluded that it was this former sire of the brown mare that was responsible for the brown stripes on the foal. And so this for a long time stood as a clear instance of telegony. Well, we now know in this case that the striped appearance of the foal is in fact due to recessive genes for stripiness. It's got absolutely nothing to do with the fact that the poor mare was previously forced to mate with a quagga. But nevertheless, in recent years, this concept of telegony has been revived, and it's now considered a phenomenon worth investigating. So this is what it is, just to remind you. So what we want to know is, how can telegony work in burying beetles? So our detective work starts with the source of the variation in larval size, which remember we alluded to earlier in the talk, because larval size at dispersal, which in turn determines adult size, is strongly related to the density of larvae on the carcass. And when there are a lot of larvae on the carcass, there are fewer resources to go round, and so each attains a lower mass by the time it disperses away from the carcass. So if we want to understand variation in adult size, what we really need to understand is what determines the number of larvae on the carcass, and that in turn means we have to understand what determines the number of eggs a female lays. And to understand that, we have to rewind all the way back to the start of reproduction, when the burying beetles first locate a carcass. And often lots of beetles will locate a carcass at once, and then they have to fight each other to determine who gets ownership. And the way that's organised is that the females fight each other, and the males fight each other, and then usually the largest individual of each sex will win those fights, and they'll pair up and they'll become the dominant pair on the carcass and take ownership of it. And then the losers lurk nearby, and they become satellite breeders. So the females become intraspecific brood parasites, and they try and sneak their eggs into the clutch of the dominant female. But the loser that we're interested in tonight are the satellite males, because what they do is they mate like mad with the dominant female. So the dominant female mates a lot with her own male, her dominant male, but she also mates with this satellite male. So the upshot is there's intense sperm competition within the reproductive tract of the female to fertilise her ova. And what we wanted to try and find out was who wins this competition for fertilisations. So Sonia and Ben carried out this experiment shown here. And what they did was they took medium females and they were given one of 4 different mating treatments. So they got to mate with 2 different kinds of males in succession. They had one male for 24 hours, we took him away, we gave him another male. So either they got a large male and then a small male, or a small male and then a large male, or a small male and a small male, or a large male and a large male. And at the end of all that mating, we gave the female a dead mouse to breed upon, and we allowed her to raise her larvae, and at the end of reproduction, we came in, took the larvae away, and genotyped them to find out who was the true dad. So the results of that experiment are shown here, and what we're looking at here is the percentage of the brood sired by the second male to mate with the female. And these are the different mating treatments. So across here, the first male to mate, and down here, the second male to mate. And what you can see is that when the larger male mates second, he gets a very high fraction of paternity of the brood. It's about 75% on average. So these large males are very good at sperm competition because they sire the majority of the offspring in the brood. But the small males have a bit of a cunning trick, because what they do is they induce the female to lay more eggs. And you can see that from these data shown here. So this is the number of eggs laid by the female, and these are the different mating treatments. And you can see that when the small male mates first, she lays more eggs than when the large male mates first. So what we think we've uncovered here are 2 different kinds of male fertilisation tactics. The large dominant males, their strategy is they're going to mate last, and then they're going to win sperm competition for fertilisation. So they get about 3/4 of all the brood being theirs. And what the small males do is they can't hope to compete with these large males. What they do instead is they mate first, but they make the female lay more eggs. So they increase the size of the pie. The proportion of offspring they sire is still small, but the absolute number of offspring they sire is larger. So it's a best of a bad job strategy for improving their reproductive success. So what's all this got to do with telegony? Well, it turns out that this stimulation of female fecundity by small males has a very profound effect on the size of the offspring that emerge at the end. And you can see that from this figure shown here. This is the average larval mass at dispersal. Here are the different breeding treatments. And you can see that when the small male mates first, the average larval mass at dispersal is significantly smaller than when the large male mates first. And in fact, we've done further analyses of these data sets where we have looked at broods where the second sire alone got paternity of the brood, so the first male got none at all, but it's the first male's size that explains variation in the size of the offspring. So even though those males are not the sire of the offspring, because of this phenomenon of fecundity stimulation, they cause the offspring to bear their characteristic body size. So it's evidence of telegony in this species. So what we've shown then in summary is that there's non-genetic inheritance of body size, and it's due to fecundity stimulation by subordinate males, which is a tactic in sperm competition itself brought about by the polyandrous mating of the dominant female. Now, how on earth is this happening? Well, we know in fruit flies and in seed beetles that males produce seminal by this means, they're able to induce females to lay larger eggs. And we've just got a grant to investigate whether this is going on in these beetles as well. And the preliminary proteomic analysis says, yes, indeed, these genes exist in the seminal fluids of our subordinate varying beetles. So, just to sum up then, what I've shown you is 3 different ways in which behaviour changes evolution. By changing ecological conditions, by being an agent of selection itself, and by mediating the way in which variation is inherited. Why should we care about any of this? Well, it matters because what it means is that the pace of evolutionary change could in fact be a lot faster than we previously thought. We can see evolution in action under our eyes in the lab in a few months. So it's an extraordinary rapid process in these beetles. And we can apply that better understanding of evolution to big picture questions that are of societal concern today regarding conservation or medicine or managing crop pests. And finally, perhaps, we can even manage our own ecology and evolution by changing our own behaviour. Okay, so I'd like to thank these organisations for generously funding our research, this marvellous team of people that I have in the lab back in Cambridge, and you for heroically sitting through all this and listening so attentively. Thank you very much, Dean.